Hi guys, my name is Katherine Atkins and I'm a neuroscience senior here at Baylor and I'm the vice president of journal clubs and bursts, and today I'm going to tell you how to read a scientific journal article. But first, before we dive in, does anyone know why we have scientific journals?
To read about science. No, I'm just playing. Research stuff like people do stuff people do research on.
Yeah. Yes, I agree with you. I agree with you. Scientific articles are really awesome because it's like a way that scientists can share their research and knowledge and advance the fields of technology and it's just even not only like scientists look at research articles, but also physicians. That's how they keep up with the times. They know, well, now this COVID vaccine should work and all of this stuff. It's it's just awesome. It's a great way for people to share knowledge and learn about something new. So the process of how a scientific article comes into being is first a lab conducts experiments and they collect data and manuscript is written. And does anyone know who contributes to writing a manuscript for a research article? Um, Well, it can be anyone. It can be research assistants, there's graduate students, it can be postdocs, PIs help, principal investigators. Um, and like, even after all of this, um, the paper usually is sent off to different colleagues in the field so they can review the article. As it says in the next step, that different co-authors and colleagues and lab members review the transcript because it needs to be perfect before it's released to it's it needs to be go through proofing and all of this so when it's so it can be accepted into the journal of which the PI or authors are submitting to. Um, so once it's submitted, the editors of the journal decide whether or not it's good enough to be sent to review, and then peer reviews happen, and this is like other people in the field review the paper, and then we have authors who edit it and send it back for more review, and then the editors make the final decision, and hopefully the paper gets published. But sometimes it doesn't get published because, well, does anyone know why a paper might not get published even after going through all these sitting back and forth drafts?
Sometimes there's research happening about maybe the same trials that that paper is going over, and if they manage to publish, or if they manage to publish before this paper does, then they don't release it. Or if they find out data that might change their conclusion, could prevent publication.
Yes, precisely. Someone else might publish before them. And also, it could just, the experiment itself might just have a poor study design. Like, there might be some confounding factors that makes the study not that strong, and they might need more data before they can publish the paper. And this is, so let me go to the next slide. Oh, so this scientific publishing cycle, it's like most labs have it. It's a continuous cycle where the article is written, it's submitted, it's reviewed, and like in labs, there's multiple times, like there's usually more than one cycle going for a different paper or project. So in a research lab, you might have one scientific paper just in the drafting process, and then you might have a mirror paper in the revision process, and you might have a mirror one just in the in the reviewing process. And like, so that's what usually goes on behind the scenes in publishing scientific papers. In a normal, like, there's many different types of scientific papers. Let's see, there's reviews, there's meta-analyses, there's methods papers that just focus on specific methods. There's editorials where like the editor will come back and be like, and give its opinion on a paper. And like, so a review is like, it's like a literature review. They basically, someone reads a bunch of different scientific papers and puts it all together into a literature review saying, this is what I learned based off all these different studies. And meta-analyses is like, there's all these different data. They take a lot of data from different databases and they analyze like different topics throughout a population. Today, we're going to focus mainly on like the normal, what you think of when you hear scientific paper. And a typical paper, it's always in the same structure. It's always in the structure. The red stuff is things that are always concluded, that will always be in a paper. And sometimes the black stuff on this page will be excluded. So the abstract, does anyone know what an abstract is?
The abstract sometimes just explains, it's like a mini version of the paper. So it'll have like maybe a sentence or two for every section of the paper.
Awesome. I would say the same thing. I was about to say the same thing as you. So yeah, basically abstracts are great for like when you're looking around trying to find a specific paper to read more about. Like the abstract gives like a little brief like summary of what the paper talks about. So it's really good when you're looking for different papers. If you, before you read the whole thing, you can just read the abstract and be like, oh, this paper doesn't have what I need. Or, oh, this paper's perfect, let me read more. Does anyone know what would be included in an introduction or background paragraph?
Can I go for it?
Yeah.
I think that you would introduce like the purpose of like what you're studying, and especially if it's like the paper is super specific, then it'll like explain like what it is and why they're researching it.
Awesome. Yeah. So if there's like any like main theories they might cover, like, or they might define some like terms, because sometimes papers get really technical and it just is helpful to have like a brief like background. And also in the introduction, they always introduce their hypothesis of why they're researching or what they're looking for behind their research. Materials and methods. This section usually just gives like enough information. So if someone else wants to come back to do the same experiments as the paper, it has enough information to tell you what you need and what they did exactly. The results, they're just like it sounds. It just tells you what they learned from collecting their data. And well, the results tell you the exact like numbers, and the discussion explains the results. It tells you their interpretation and what it means. And usually the discussion conclusion kind of mix together in most papers, but the conclusion can, if I was to separate it from the discussion section, it would like give implications of what can be done in the future, like different points of study. What would be the next step if the study continued? Citations is important. Supplemental information, that would be more so like different, like your graphs and like any imaging or anything like that. Extra stuff. So we covered that. So how do you go about finding a research article to read? Well, so the best thing, usually the title will state like what the researcher's researching. So when you're approaching how to read an article, if you don't recognize any like key term, if you don't understand any words in the title, just Google them so you know what it is. Also, you want to read the abstract. If there's any unfamiliar terms, make a list. You don't have to Google those terms right away, because maybe in the reading the introduction, they'll answer the term, they'll tell you what the terms mean. And if they don't, you can always just look it up. And also another strategy that I like to use, especially when it's like really biochemistry stuff, I like to like diagram like the different like pathways, like just so I know what's going on. Or like I'll diagram, well, they did this, then they do this. I would make like a little flow chart just so I know. That's ways I like to do it. Methods in general, when you're reading the methods section, you don't really need to have it get into the nitty gritty details, because you're not replicating the experiment. But you want to understand what they did and why. So why did they decide to use mice over cells? Just like different factors like that. Usually you can ignore the results section, because they'll just be, because the discussion will go more in depth in explaining the implications of the results. You can look at the charts, and the charts will like help you understand the discussion section more. And looking, and you want to have a broad understanding. It's good to have a broad understanding when it's coming to reading articles, I feel like, over just like a single little fact, although there are like significant facts, but usually you want to have a broad perspective when you're looking into reading articles. So I just basically told you just now, I just like said like the general, how you would, if you had like a lot of time on your hands, how you would go through a paper. And basically, so you start off with the title, Google things you're not familiar with, read the abstract, and then if there's terms, this is if you have a lot of time, the intro, how many, you read the intro, and then the methods, just you want to have a general interview and all of that. Does everyone follow me? Basically what I was talking. Sweet. So let's try it. Okay. So when you're looking for a research article, you want to find the right article. So this is back to how that I was talking about, how the abstract is really helpful in determining if the article is the article for you. So there's different databases we can use. PubMed, it's really good. When you search through PubMed, it has these key terms usually, and it looks for, if you type in a word, like let's say caspladin in mice, then it would like specifically look for the words like caspladin in mice. It would look for caspladin in like the title, and the abstract, and if it has a little keyword section. Sometimes articles have keywords. It won't look for it won't look through the full paper if it's talking about caspladin. Now, in papers, usually with PubMed, they suggest more reputable sources. Google Scholar is a more broader database article search site. It will have when you type in like keywords, once again, let's use caspladin for example. If it talks about caspladin like one section in the discussion randomly, it will pull up that paper. It looks through the whole article instead of just analyzing the abstract and the title and any keywords. And it also pulls up journals that aren't as highly that are more sketchy. It'll bring up some websites, so they're not as reputable as PubMed websites, but it's more broader scope. So, and then also Baylor libraries. We have OneSearch and all of these cool research as well that are free to use. So it's really nice. Also, when you are looking for an article, and let's say you found an article, right? And you're like, okay, this article, it talks about caspladin, but it doesn't really talk about it being used in the way it's not really comparing it to fatigue and motion, what I was really interested in. You can sometimes if you find an article, you can go down to the reference section, so where all the citations are, and maybe the article you're looking for, it will be down there. If there's like one random sentence that's like cited in the background that you really like to learn more about that study, well, the citation at the bottom of the page will take you to the original paper. So that's really helpful. Now, let's say you don't have a lot of time and you're just wanting to skim it. The method I suggest, and also by this paper down here, one good way to like skim it and still have a really good idea about what happened in the article is to you do this 3-pass method. So in the first pass, we read the title, the abstract, and the intro really closely. You skip the methods, the results, and graphs, and go straight to the conclusion discussion section. And then in the second pass, you read it again, just those 4 sections, and then like is verbally yeah. So see, does this share when I go like that? Share. So this is where I got this from, but it basically breaks it down even more if you're really interested. I can send you a link if you email me to this paper, but basically in the 3 steps approach, questions you want to have in your mind when you're reading is, what type of paper is this? Usually it's going to be a research article or maybe a literature review. And then like what's going on, and then the context, what's the problem, what techniques were used in the problem. These are just questions that are good to have in your head when you're reading. And like also, does it make sense what they're going after? Because in all research papers, they always like have this introduction, and they'll like cite like different experiments. Well, they should cite different experiments supporting why they're wanting to do the experiment, what's leading them to conduct the experiment. And then, and also it's well written, pay attention, blah, blah. So yeah, that's basically that paper. If you're really interested, I can send that to you. Let's go back to the PowerPoint. Okay. If any of y'all guys have questions, feel free to stop me at any point. Okay, so in conclusion, the skim search, you always want to read the abstract, the intro, and the conclusion. And if you have more time, you want to maybe read the methods for like general schemes, and like look at the graphs and diagrams and the discussion. Unless if you're trying to reduplicate the research or replicate the research done, you don't really need to focus hard on the materials and other details of the section and the results and all of this. So you don't really need to focus on this. You just mainly need to focus on this when you're reading a paper, just to gain knowledge about what's going on in the paper. Do we have any questions? No? Okay. I wanted to do a quick, like, brief understanding of different statistics that are used in just a quick intro, because I feel like this will help you when you're reading research articles, just common things about stats that are commonly used. Whenever you see an N like this, it refers to sample size. So that's basically how many people were in the study. The mean is just the average, and the standard deviation has this symbol. The independent variable is I, the researcher. This is what I'm controlling. And the dependent variable is what they're measuring. So example of that would be like if I was watering plants, I would be measuring how tall they are. That would be one random example. Does anyone understand what the standard deviation is?
Yes. It's the scores that vary around the mean.
Yes. Awesome. So standard deviation, it goes back to, it has to occur in a normal distribution, or the bell curve, as they like to be called, as it's also known. So this is our mean. So the average score is, think of like the SAT and ACT. They do this too. They bell curve it. So the average score is this, or the MCAT. So the average MCAT score is always bell curve. So it's 500, and then up. So the standard deviation is how typical the average deviance from the mean. So within one standard deviation above and one standard deviation below, that's about 68%. And then you have 95% within 2 standard deviations below and 2 standard deviations above the mean, and close to and 99% for like that. A mirror way that that is shown to like represent the distribution and basically scores is a box and Worcester plot. Has anyone ever seen this before? So basically in a box and whiskers plot, so it's broken down into quartiles. So think of class rank. So you have like your first quartile, your median is your second quartile, and your third. And the box area is shows in between the first quartile and the third quartile. And then there's this and that's your IQR, your interquartile range. Now, and then from the box, there's these whiskers that are your fences, and you just go 1.5 below the first quartile, that's your lower limit, and then 1.5 quartile above, that's your upper limit, and anything past that is your outliers. So an example of like, this like applies to other things other than just box and whiskers plots. Like looking at this article, that's why you see this like line. This shows the interquartile range, and this is the box. Does anyone know what these asterisks are on the graphs? See these asterisks, these little stars?
Do they refer to something at the bottom that's down there, or they're referring to some sort of, I'm not sure what it is, but.
You're good. It refers to this down here. It says these are p-values. It measures significance. So the more little asterisks, the more significant through stats it's shown. It's really significant. Here's just a mirror image. Sometimes they will show each data point as a dot. So a mirror way to compare significance is knowing where the little bars are. We can say like if they overlap with each other, we can say, well, this is not significant from each other because they're overlapping. But over here, you see there's like a gap in between the 2 bars where so like this would be significant. And as shown, there's little stars. So does anyone know what a p-value is? Yeah. So p-values go back to confidence intervals and tests, which are 2 ways stats are used a lot. In a confidence interval, it's used to predict the true stats within a range of the alpha. Does anyone know what an alpha is in terms of statistics? No? The alpha is basically the percentage of like, it represents the percent that is expected outside of the research range. So like, an example of a confidence interval would be like, we are 95% confident that that there is 1% to 5% in people of this region that have COVID. So that means like the 95%, so it would be a 5% alpha, and the 95% would be your statistic that like, so like 99.5% of the time we are expected to that the average number of COVID cases is between 1% and 5% in this area. So that's a confidence interval. Does that make sense? Does anyone have questions? Okay, good. Tests, there's many different types of tests. There's the CHI test, there's all of these different types of tests, but they all boil down to there's a null hypothesis and an alternative hypothesis. The alternative hypothesis is what the researcher's trying to prove, and the null hypothesis is basically the opposite of the alternative hypothesis. So for example, let's say my alternative, I'm guessing my score on my last test. And I say that my score for my alternative hypothesis, I predict that my score is equal to 85. I got 85. So my null hypothesis would be I got something below 85 or higher than 85. But of course, it doesn't always have to be equal to a certain number. It can be, I believe I got higher than a B. I got 80 or higher. So I would, if my alternative hypothesis was wrong, then my null hypothesis would be anything less than or equal to a B. You can use less than, greater than, equal than, and all these hypotheses. So going back to the p-value, what was the asterisk stuff? So basically, if the p-value is smaller than the alpha, so the alpha, you remember the 5% that we're unsure, the percent of time it's like not correct. Well, if it's smaller than the alpha, then the researcher rejects the null hypothesis because the results are statistically significant. Now, if the p-value is larger than the alpha, then the researcher cannot reject the null hypothesis because there's still a chance that the null hypothesis is true. And since there's no evidence that the alternative hypothesis is 100% true, we can't say the results are statistically significant. Does that make sense? Does anyone want to say it back to me in their own words? No? Okay, you don't have to. Okay, let's go on. Okay, so test statistics, you might come across when you're reading all these different like letters, and there's so many more. I just did the basics, but whenever you see a Z statistic or a Z test, that's usually used when comparing populations, so different like samples. Now, when you hear like a T test or a T statistics, that's used when they're comparing means. So that would be like for my null hypothesis, for my alternative hypothesis, I believe that the average for the class on the test was a 70, equal to a 70. See, that would be an example of a T test. Now, a D statistic is still looking at two means, but it's looking at how far apart. So an example of this would be like, I believe the difference between my class average and the other section's class average on the means test. So I believe there's like a 6-point difference between the two classes. That would be an example of when you would use a D statistic. A chi-square statistic is used when comparing distributions, specifically categorical distributions, so like colors or like ethnicity, race, male, female. It would be used in like those situations when there's like multiple like factors that are not really quantitative, they're more qualitative. R squared, that refers to it's also, there's beta and all this. This is all refers to linear regression. So basically, it's like they have a line and they know, okay, how well do these points, our sample points, fit to our line of fit? That's basically what this statistics testing. And an F statistic is used in an ANOVA test, and this compares multiple means. This one only compares like 1 or 2 means. This one compares like multiple means. So that's just there's more test statistics. I just put common ones that I come across down right here. One last thing I want to touch on before I end this is type 1 error and type 2 error in power. So type 1 error is basically a false negative. That's when we say the null hypothesis. That's when we say we reject the null hypothesis, but it was true. So, and a type 2 error is a false negative. So that's when we fail to reject the null hypothesis and it's false. So something that helps me remember it is this image. Basically, our type 1 error. So in this example, does anyone want to tell me what our null and our alternative hypothesis might be in this example?
Would the null be the you're not, and then the alternative be the you are?
Yes, you're correct. So a false so whenever I think of type 1 error, I think, okay, it's like it's when you're telling the patient, they're using what our scenario was, that the null, it's you're not pregnant, and the alternative you are, you would be telling someone they were pregnant but they weren't. And a false positive would be a false negative type 2 would be you're not pregnant but you really are, you're being told that. So from the two, which one do you think is a worser error to have? A type 1 or a type 2?
Type 1, maybe?
Yes. So, yes, type 1 is, wait. Well, actually, it probably depends on the situation, but I think in that case, yes. So another thing that researchers are really, like, when they're talking in their paper, they always want to increase this thing called power. So power is basically is when you have a false, the null hypothesis is false and you reject the null hypothesis. So it's the correct decision. There's different ways to increase power. So like, this is why it's always important, or it's always good to have a large sample size, because this normalizes the bell curve we were talking about earlier. So having more people in, we have a closer, it moves the mean to be more exact. So that's why researchers always want a large sample size. So let me pull up this, because I think this is cool and it helps me understand it better. Sure, okay. So normally, let's say you don't want to change, researchers don't want to sacrifice their alpha, because you can have 100% accuracy. You can have and say that I scored, I know my test score is anything between a 0 and 100, but that doesn't really tell you much about anything. So it's not good to have a really low alpha that's like actually 0. You usually want to have like 0.5's the normal one. I'm going to say the power is. So when we're trying, so this is a good demonstrator to show you. So let's say why increasing the sample size helps with power, increased power. So right here, I don't want to sacrifice my alpha because it's meaningless, as I said before, if we do. But of course, we don't want to have it too big of an alpha. Usually, it's either 0.1, 0.05, or it's either 10%, 5%, or 1%. That's the normal alphas. Usually 5% is used the most common. But as you can see, this shaded portion right here is the chance of creating a type 2 error, which that's something we do not want. So if we increase the sample size, and this is our beta right here, when we increase our sample size from 20 to, let's say, 40, you see we have less, the means come, we have less, you see how like it makes a more of a arch shape. So like it's more normalized, and in the alpha, the beta type 2 error shrinks. So even if I jump it to an even bigger sample size, there's less, or let me put this, there's less, well, I set the power to be equal. Let's change it to alpha, or no, sample size. But basically, it shrinks. The type 2 shrinks when you have a bigger sample size. The bigger sample size, the more it shrinks. I don't know why it's going the opposite direction, but yeah. Oh, you did this in stats class today? That's cool. Who's your professor? Oh, anyway, but that's basically, so those are common things you might come across in your research paper. Does anyone have any questions for me? No? Let me end this recording real fast.